All right, we're going to talk about the lectin complement pathway next. And this pathway is a little bit different from the classical pathway. If you recall back to the classical complement pathway, it starts off with antibodies binding antigen on the surface of your pathogen. The lectin pathway does not use antibodies, and so it's said to be more of an innate immune response. So instead of antibodies recognizing antigens, what it uses is an initiator protein in the pathway recognizing carbohydrates on the surface of your pathogen, such as your bacterial cell. So you can see here I have some carbohydrates on the surface, and the types of proteins that usually recognize these are what we call lectins. In this case, the initiator protein is going to be called mannose binding lectin. And we're going to see that it binds directly to those carbohydrates. So it actually has this stalk region here. That's a triple helical collagen structure. And then the carbohydrate recognition region of this protein that binds the carbs. So we call this MBL, or mannose binding lectin. And it's one example of a protein that could be found in this lectin pathway as initiator protein. There's also another type of lectin that you might hear about that's called ficalin, which is a very similar protein that has the same function. It binds carbohydrates to initiate this lectin pathway of complement. But we're going to look just at MBL today. So MBL actually associates with serine proteases. You can see it has a very similar function to the C1q-R2-S2 initiator complex in the classical pathway. If you remember, that one associated with C1R and C1S that had serine protease activity. This mannose binding lectin associates with proteins called MASPs, M-A-S-P. We have 1, 2, and 3. And it's been shown that MASP2 is the one that's going to confer the serine protease activity of this whole initiator complex. So MASP2 binding to MBL start the lectin cascade in this complement pathway, or the cascade of the lectin complement pathway. So what we'll see next in this pathway is after our initiator protein binds the carbohydrates and we have our MASP2 serine protease activity, we're going to basically have the complement protein C4 come in. C4 will be cleaved into C4b and C4a. And if you recall from last time, C4a is one of those inflammatory mediators. C4b will be associated with the membrane. And then everything else is basically the same as the classical pathway from this point out. So really just that initiation step is different in the lectin pathway. You know, we rely on carbohydrates. This initiator complex is called MBL or ficalin, binding those sugar residues, the mannose on the bacterial cell surface. After that, we've got C4, then we have C2 coming in. So C2 is going to be cleaved into C2b, C2a. C2a is the one that comes down here, whereas C2b diffuses away. So we've got C4b, C2a. This, if you recall from classical pathway, forms our C3 convertase, or you might just hear it referred to as C4b2a for short. So then the next thing that would happen is C3 is acted upon by the C3 convertase. We make C3a, which is our inflammatory mediator. We make C3b, which comes down here, associates with C4b and C2a. And now this entire complex is referred to as C5 convertase. So this should again all be review for you from the classical pathway. So C5 convertase, sometimes also abbreviated C4b to A3b. Remember, we also make other molecules of C3b that can serve as an opsonin to coat pathogens and allow for increased phagocytosis. So now the C5 convertase is going to act upon C5, shown here. We're going to cleave that into C5a, the smaller subunit, which is an inflammatory mediator, and then C5b, which goes next to the membrane, and that's going to start the formation of our membrane attack complex. So it recruits C6, C7, and C8, and then finally 10 to 19 subunits of C9. And that all forms a large hole, a cylindrical hole in the membrane, making this structure called the MAC, or membrane attack complex, which results in loss of integrity of the membrane of the pathogen and cell death. So that's a brief overview of the lectin pathway of complement. You can go to the Google Sheet now and do the review activity. We're going to model this yourself online. Please let me know if you have any questions as you're working through that.